morning, everybody. We are going to talk a little bit about study design. We are at the stage where you have an idea that you want answered with metabolomics, and this is a point where I emphasize that consultation is a great idea with the core, because some of these questions can be answered in a very effective manner if you sit down and have a discussion with the core and tease out the specifics that metabolomics requires. I'm going to touch about some of the points that are required to be thought of before a design of a study is done, but a lot of these topics will be dealt with other speakers down the road. So in this topic, we're going to talk about how your biological questions can be answered by metabolomics and what are the factors that you need to think of. We've seen this probably many times already this day, but you have to understand metabolomics is a downstream effect of what genes are transcribed, translated, modified, and then are the end effect of enzymatic actions. Not only that, they're also the effect of external factors like diet, your age, your lifestyle, what drugs you take. So that being the basis of my talk, I'll proceed on further. We'll talk a little bit about the different model systems that you can use to ask your questions, what biological materials we can analyze, what study design and power that you can consider. Study design alters the metabolome, so that point will be reiterated again and again, and what strategies we can use to prevent these variabilities. To start with the model systems that we can use, cell, animal, and human. A cell metabolome is very basic and very easy to handle because it's a very controlled situation. You need small replicates because of the control situation, and we can study both the endometabolome of the cell and the exometabolome as the secretome of the cells. So as I mentioned already, they are reproducible, need small numbers, and also very easy to study the flux of metabolites in the system by labeling your media with isotope labels traces, basically, which have a small mass difference to your regular metabolites, and tracing them into the cell and detecting the metabolism that you want to study. Animal studies are also quite compatible with metabolomics experiments. You are reducing the inter-individual variations because you're growing your mice or other animals in a very controlled situation. And usually they're from the same genetic background, and because genes influence metabolites, this becomes very compatible for metabolomic studies because the only difference could be your intervention or the disease phenotype you're studying. Also, as we all know, animals, we can harvest simultaneously many tissues and biofluids, therefore we can trace the metabolite through the entire living system. Human studies are the more common metabolomic studies, but also they are marked by inter-individual variations. Sometimes diet and environmental factors are not under our control, but it's also a very well-studied group of organisms. Therefore, there are nearly 4,000 quantified metabolites that are published out in the literature. Therefore, they are standards to refer back to. So what biological materials can you study? Pretty much everything. The cell system, we require about 1 million to 10 million cells. We can study the culture media. We can study biofluids like plasma, serum, urine, saliva, CSF, pleural fluid, ascitic fluid, also different kinds of tissues, skin, nerve, organs, and fecal material, because the latest trend is to be interested in the microbial metabolome that influences the human metabolome as well. The specifics of metabolomics is such that you require very little sample material when compared to other technologies. Therefore, it's a great advantage to metabolomics. What different study types are good? What are the pros and cons? The very basic study design type is a case control, where we use well-matched cases and controls to discriminate metabolites between cases and controls. Other than having excellent correlation to disease, they can help us detect altered pathways and have a huge biomarker potential if they are powered to detect the differences. But they're also prone to biases because you cannot differentiate cause and effect. But also, there's a lot of chance to induce study design artifacts. For example, if you're studying heart disease and comparing them to controls, the biggest difference could be drug metabolites if the heart disease patients are on statins. So these are artifacts that are introduced because of study design. And if you have a well-documented cohort with well-matched characteristics, we can tease them out beforehand or afterwards with statistical inference. The next study type is the interventional study type. This is great because you are producing a pre and post effect. Therefore, with a same individual, you can detect the intervention differences. Also, we can detect time series and therefore comment on the stability of the metabolite you're studying for future biomarker potential. Crossover studies are even better because here you're kind of minimizing the inter-individual variations that normally human studies have an issue with, because the same patient can be his own control, therefore becomes very tight and the power of the study increases. Sample size. This is often a very important question before thinking about metabolomics experiments, and also very important because you want to place them in grant applications and you want to make sure you're not wasting money if it's not an adequately powered study. Deciding on the power of a study is pretty generic for other parts, but except in metabolomics, we are measuring thousands of variables, and that's the only difference that we need to consider. Because you're measuring thousands of variables in the untargeted approach and also a very large panel of things in the targeted approach, this increases the false positive rate, that is chance of increasing the type 1 error in studies. This is dealt with statistically at the very end part of it by increasing, doing what we call the false discovery rate. Therefore, we apply more stringent criteria for achieving significance, which means that you have to increase the sample size more to achieve the same significance that you would have achieved before. Also, this can suppress some valuable data because of your more stringent cutoffs. So, a priori knowledge of effect size does not exist for most metabolomic studies. So, the initial strategy could be to study a small discovery cohort, which is well matched to study what the effect size of the metabolite in question, and then design a more highly powered validation study, which is well powered. Biological variability that I briefly mentioned in human studies, it's a huge problem because most clinical studies are gathering samples over time, over seasons, and some things change over time, and therefore there could be problems with short-term and long-term variability. For example, if you're measuring vitamin D, over seasons there could be huge differences in your cohort. So to determine and control for these things, you need to document the time of sample collection. Therefore, we can make sure that those things can be eliminated in analysis. How do you plan for data normalization? Because when you want to measure something, you want to feed the same amount into the analytical system. Therefore, we need to consider some of these things even before the analysis is done. For example, in a cell system, you would want to know the exact cell number that you seeded into your plate. You could also take into account the volume of certain biofluids as a means of normalization. Concentration of certain key metabolites, for example, glucose in plasma, could be one to normalize things across the board. Urine is particularly prone to these problems because of concentration mechanisms in the kidney. So we nephrologists typically use creatinine, which is a constant metabolite that is produced in the same rate every day. Or you can use osmolality as a measure of concentration to normalize things. For most tissues, we use protein concentration as a normalizing factor. Some things like feces might require a dry weight estimation so that you can normalize across the board. If all these things have not been thought about, we can still normalize using a total ion chromatogram, that is the electrical charge that the machine sees of the entire samples, as a normalizing factor. What are the factors that affect the metabolome? At this point, I want to reiterate the point that we are taking snapshots of the metabolome. We are not capturing the dynamic nature of the metabolome. Therefore, timing your sample collection and timing your study design is very, very important. I'm going to show you a few examples of the fact, and these things will be elaborated further down the day. For example, in cell metabolism, seeding densities matter, passage number matters. These all determine the proliferation capacity of the cell. Therefore, will change hugely between sample conditions if you don't do a consistent manner. Growth conditions like temperature, ambient atmosphere, and culture media quantity, and also the quality, what metabolites are in the culture media like glucose and glutamine, determine the exact nature of your cell's metabolism. Here in this example, this is myeloma cells that are being grown. We are measuring glucose and lactate across time, as you see. As you see, depending on what time point you capture, the cells are using up the substrates that you provide and producing lactate. So depending on which time point you choose, you will get a different picture. So you have to standardize your collection time points across the board. This is another example where 10 volunteers were made to stay awake for 40 hours. Here we are measuring the precursors of steroid metabolism, the long-chain fatty acids, across time. And as you see, with time, these metabolites change in concentration. And depending on where you measure them during the time of the day, you will get a different picture. So circadian rhythm here plays a role. So this is 24 hours repeating itself again. So the sample collection time needs to be standardized. This is an effect of feeding, fasting, and activity and stress on different metabolites. In this particular example, this is 15 volunteers that we are measuring free carnitines and acylcarnitines over the time of day. This is fasting phase, 24-hour fasting. You can see how the different metabolites of carnitine change. And depending on how many hours of fasting you do, you'll get a different picture. Because if you sample here, the picture is different to sampling here. So even fasting time needs to be standardized. Here, it's a special liquid diet that we're given, and you can see the differences in the carnitine levels. This is a glucose tolerance test, and see the differences. Here, this is physical activity, 1 hour on a treadmill. You can see the huge differences that creates. Here is a lipid tolerance test. And here is cold stress. You simply immerse your hand in 3 minutes of cold water, and that's the difference the metabolites are creating in the plasma. So I hope I convinced you here that all these factors are influencing in metabolism and therefore need to be considered in study design. This is an example again of 8 volunteers who have fed the same 3 different diets over a 4-week period, and the urine was sampled over different time points. This you can see in this PCA plot that depending on the diet, which is saturated, monounsaturated, polyunsaturated diet, the extreme changes that this diet is causing in the urine after a few hours. So again, diet is important. Diet histories are important, not just diet, medication. If you time your sample collection near key medications, for example, insulin, it will change the entire metabolome in a matter of seconds. So what are the effects of different sample collecting and storage strategies? So again, I'll have a few examples here, and Kelly will elaborate in the next talk further. This is the effect of serum versus plasma. Here is plasma in an EDTA tube, and this is serum. You can see that the chromatogram is not exactly the same. For the most part, there is acceptable correlation of nearly 104 of the 121 metabolites, but some key amino acids, phosphatidylcholines and lysophosphatidylcholines do differ. So if you have a study that you started off with serum and then you don't have some serum samples, so you're deciding to use plasma, you have to think critically what you're trying to measure. Are they correlating with each other? My advice is to stick to the same kind of sample. Also, the site of sample collection, for example, did you get an arterial blood versus venous blood? Is it carotid artery versus femoral vein? You know, all those things make a huge change because of the way the metabolism is very dynamic. Also, different sample processing strategies could have huge effects. For example, if you decide to quench your metabolism with various solvents, this is liquid nitrogen versus solvent at -80 versus solvent at 0. This is a glycolytic pathway. As you can see, same samples with different quenching mechanisms have different metabolites, causing significant changes. Similarly, whether you decide to scrape versus enzymatic digestion, centrifugation speeds, hemolysis of plasma and serum samples, All these have changes to what you're trying to measure, so need to standardize and be consistent. This is the effect of bacterial growth on urine. This is urine left on the bench for 24 hours using an NMR spectra. As you can see, this is a normal urine sample. 24 hours later, new peaks, trimethylamine peaks are forming. So one effective strategy for this is to add sodium acid into urine containers so that bacterial growth is prevented. Other effects are effects of temperature. This is the effect of temperature on hypoxanthine and sphingosine 1-phosphate. The best strategy for this is to work on ice and limit processing time, transport on dry ice, store in -80, and limit freeze-thaw cycles, which also decrease your metabolite concentrations. So I hope I've convinced you to time your sample collection and be consistent with the timing of the sample collection, standardize diet and medication history, document all sample-related information so that you have that available for statistical interference later, standardize collection strategies, and keep them frozen. So in conclusion, a good metabolomic study design will have a well-documented, well-considered study pattern and have a standardized protocol. And if you got anything out of this half-an-hour lecture, I hope it is consistency and consultation with the core. And these are my references. I can take some questions right now. Yes. Mm-hmm. Mm-hmm. Yes, we measured. So as you probably recall, glucose is, we also standardize by fasting. So do we with cortisol. We also do the supine standing point to cortisol as well. So these are not new differences. It's just that when you measure a large group of variables, we tend to figure out that, forget that there are things in this group that will change with what fasting state or feeding state or activity state you are in. So these are not new. These are known things, but people tend to forget because you're used to proteomics and transcriptomics being relatively stable across a time, but metabolomics is more dynamic and it has a lot of biases introduced by other things. That's the only point I was trying to make. These are not new. They are facts known before, yes.